We are making the fluorescence sensors, and we are using this Doppler as our approach. Doppler is we use the strategy as a diversity, and fluorescence as the modality that we make them. So to give you some idea, here's one problem for you. This is cell culture from brain. Take out the brain and then grow those cells. Inside this different type of cells, there are neural stem cells. Can you find those neural stem cells? I started as a chemist, so when I studied this biology, if I look at those cells, just the black and white pictures, time to time, even that's very difficult to find which is a nucleus, which is a mitochondria, something we learned in the primary school or the biology. So finding out those specific cell type from these mixtures of cells is very difficult. So I dreamed some way, if you make some probes or some sensors, okay, this sensor, if it goes into the cell and if it shows one type of cell, so this is a neuronal stem cell. So we wanted to develop this kind of fluorescence probes. That's our goal. Then as a chemist, how do you make this kind of other probes? So one very possible other way, and also the conventional way, is you make this kind of a sensor or probe, and then usually it has two motifs. One motif is a binding motif, one motif is reporter motif. And this binding motif binds to its target. And if it binds to the target, somehow it's changing its colors. And then that color change is used as a fluorescence sensors. That means you know your target from the beginning, and then somehow you can design of those other probes. Then this approach we can call target-oriented approach. Sometimes you can screen them, and then you can find out the best antibody which bind to that other target, and then label with the fluorescence molecules. And sometimes you can use the peptide or the aptamer. But what if you do not have any idea about your target? Still you wanted to find something. Then one possible way is we make these sensor molecules as toolboxes. And this sensor molecule has internal flexibility. That's why it can absorb the light, but it loses the energy by vibration. So that's why it is not fluorescent in the beginning. If it binds to the target, then this binding fixes this rotation, and then this whole molecule becomes rigid, and then this molecule has a much better chance to be fluorescent. So this is our idea, how to prepare this kind of molecule. And when we started this project, that time I was in New York, and we were looking at those all known fluorescence molecules known to the human beings. Currently, we have about 4,000 molecules in molecular probes. In Vitrogen, LifeTech, now Fisher, the Thermo Fisher, their company has about 4,000 of these compounds. But core structure-wise, not so many core structure has been known to the chemist. About 10 very popular ones: coumarins, fluorescein, rhodamine, and cyanins. So this is fluorescein. And this is rhodamine, which is you are using for the highlighter for making your the writing more visible. Now, if you have this carboxylate here, it doesn't rotate here. That's why this is a rigid molecule, very fluorescent. Our idea is, if you remove this part and then it starts to rotate here, then this molecule is quenches, like down to 10% or 1% of fluorescence. Only when the target molecule comes in bind to this molecule, it can potentially become fluorescent. So using this very simple idea, we tried to accommodate all known fluorophores to the human being, tried to build up this library. It took a long time, about 6 or 7 years since I moved to Singapore. We made many, many molecules. I said many, many. How many? Can you guess? We made a little more than 10,000 molecules of this set, all purified and then all characterized. So maybe this is arguably maybe biggest collection of those fluorescence molecules. And last year we celebrate, at least color-wise, we covered all the colors from the blue color to the long red color up to the near range. Now the next question is, using this library set, can we find out those sensors, whatever we meet? Like an antigen we have into our body, then can we generate every antibodies for them? So that is the quick idea. So first step is, let's say we have some known other target, let's test them. But 10,000 is not trivial numbers, and 10,000 molecules, if you put them into the 96 well the plate, so you have about first column, last column empty, and then 10 by 8, this is about 80 compounds. And then this kind of a place, you have a 150 place. It's still a lot of the material you need to test. You need to think the fast way how to test. So cheapest way we found so far is using this kind of a camera box. Black box with a UV lamp, and you put Canon camera on top. and regular digital camera, and then take a picture before and after you analyze the edit. This is the cheapest way I could think, about $2,000. If you use a cheaper camera, maybe less than $10,000 or $1,000. And of course, you can use this kind of plate reader. You can get this kind of a spectrum. So let's test together something. How about this? Coffee. In coffee, there's one important ingredient. What is it? Caffeine. Yeah, maybe that's the only ingredient you know. Yeah, caffeine is the most important ingredient in coffee, which can make us awake. So activator for the brain. But sometimes some people cannot take this caffeine too much. Pregnant lady and also the autism children, they should not drink too much caffeine. It's not recommended. But if you go to the Starbucks, if you buy those coffee, you can order the decaf coffee. These days, the decaf coffee technique is so well developed. It's less than 5% or less than 10% of caffeine, but taste is almost similar. Now, it's very hard to say whether this is really decaf or regular coffee. So if your goal is bring some machines and then find out this caffeine before you drink, okay? Then one way is using this camera method with and without the caffeine. No caffeine, with caffeine. It shows a strong orange color, and then you can confirm by this dose-dependent curves and selectivity test. Then yes, this is a kind of a CanCom type of material you can publish, but we wanted to go further. You don't want to bring this kind of a spectrophotometer to the Starbucks and then test it or extract. So we may spend another half a year to develop further how we can use this on-site within 1 minute, and then you can use very simple machines. So our final sensor is this green laser pointer. And then using this, if you read it, if you see this strong or the red light, this is a regular coffee. If you see this green light, this is decaf coffee. So this level we want to develop, these sensors, which can be used in real life. And then how do you know whether this is only Starbucks coffee? And we tested the Starbucks and Coffee Bean and also Singapore local coffee. Singapore local coffee means very strong, no flavor. It's a so-called coffee of kosong. And very strong coffees, any coffees, you can see this red color. How about the other coffees? Milk, sugar, cappuccino. All of them shows a strong red color. Any kind of decaf coffees, they show this green color. Then you can say, okay, by colors. If it is red color, stop. Think about it, whether you can drink it or not. Green, you can drink. Kind of level of practical sensors we want to develop. So we applied this into the different problems. Some of the biological problems, DNA, RNAs. If it's a DNA, then you can stain the nucleus. If it's RNA, RNA is more difficult than DNA. But RNA case, we can show this nucleolar spot, which is the center of ribosomal RNA synthesis. You can find the others, G-quadruplex or the proteins. One of the most abundant protein in the blood is a human serum albumin. And this protein is binding to the most of our drugs and affect those the drug property. And it is known to have two binding sites, site 1, site 2. And depending on those drugs binding property, drug has a different kinetic problem in the body. That's why figuring out where do they bind is quite important. And then to make it very easy, we found one company developed one other sensor which binds to the site 2, which shows orange color. Site 1 is a blue color. If the dyes bind to both of the sites, then it shows white color. If it's the drug, replace one of them, it shows that site 1 drug shows you the orange color. Site 2 binding shows you the blue color. If it binds both of them, it's a black color. Okay? Using this simple test, then you can test literally hundreds of these the drug candidates or the drug compounds very quickly by looking at those colors. White color means it does not bind. Black color means it binds both. Site 1, site 2. It's very easily you can utilize this for high-throughput screening. So we expanded this into different type of materials. Amyloid plaque is the Alzheimer marker. And then you can test this in vitro first. And then use them into the tissue and eventually in the live animal. And then you can inject them and then you can see those plaques on here, and then you can see those plaques. So can be used, some sensors can used in biological systems. Expanded to other things, small molecules, and glutathione is the reduction markers, oxidation reduction markers in the cell. And caffeine I showed you. One more example. If you go to the discotheque, Singapore is quite the safe country, so not much other bad things that happens, but some countries, advanced but some diversity of the people, sometimes they are the bad people, they can spike something funny in your drink. And you want to know whether it is safe or not to drink. but maybe your guy is the good guy, so you don't want to do the experiment in front of him. So that's why maybe some easy method, again, using this the green laser pointer. If you see the green color, that's okay, you can drink. If you see something funny, other colors, then you should not drink. So this kind of drink maybe you can call, maybe date, green date. And then now I didn't say about those the mechanism of this work. We found a very interesting mechanism here. Many of those fluorescence molecules, though they are hydrophobic, if you spike them in the water, they are aggregate and self-quenching. If those analytes goes in and interact, relatively molecularly, structurally interact, then they can smear in, make this bigger and bigger, and then fluorescence is becoming the stronger and stronger. This is so-called disaggregation-based the sensing. If you add large amount of the organic solvent, of course, it shows you very high fluorescence. But in the balance, if you add a reasonable amount of them, it's quite selective to that analyte, but it shows this kind of fluorescence increase, which has not attracted the people's attention. But over and over, we found that this is quite general mechanism. So we made the big, extensive review paper, and in the Chemical Society review, you can find it if you want. One more example. So using this mechanism known, we thought, how about the milk? Milk is quite important nutrients to, of course, their baby, but human being also utilize the milk a lot for bring our own babies. Now, some countries, they need to check this milk quality. For example, India. India is a technologically very well-developed country, but this milk business is many people live in the rural area. They don't have access to these specific machines. And then just collect the 10 people's milk and then sell to the 10 people's the consumers. It's kind of a small scale of business is more than half. And then their measurement of this milk quality is measuring the density. And then there's a lot of spiking of different materials, making it a higher volume kind of thing, some adulterations. And also maybe the quality of the milk. The primary criteria is measuring their fat contents. And measuring this fat contents is quite important. And we targeted to test this fat contents of the milk. And then if you add this dye, it's quenched in the beginning. And then if you add this milk with the fat, then fat goes inside here, and then it can make this fluorescence. Utilizing this fluorescence, and then we found that this aggregation is slowly disaggregated, and we confirmed that by the NMR, and also by this the experiment showing that NMR shows this the increase of those the disaggregation, and then shows this the fluorescence. It is quite linear to different. This is the size of those particles. In the beginning, it's a small particle. If you measure those scattering, becoming bigger and bigger and bigger. If you add much larger, and then it break. Nanoparticle goes away, and then it's a saturated signal, you can see. And this one is quite linear to the lipid contents. And interestingly, it is not bothered by a similar percent of protein or the sugars. Only it responds to the lipid. And then you could buy this the milk from shops, different company the milk in Singapore. And then all of them matching very well to this. But to bring this machine again, you cannot bring these heavy machines to the cow and then take the milk and measure one by one. You need the electricity source and a typical problem. So we are trying to make this into the current method is utilizing this sulfuric acid and then adding showing the height of those fat. We wanted to make this one into the portable machines. So far we have this size. We wanted to shrink down like this size and then carry on, and hopefully can help some the people that are in need. So I showed you some examples of that we have done so far. Now, this one, it doesn't have the limit because our sensor is non-biased sensors. If you have a clearly known target, then it's very doable. If it's too many targets, it's not a problem. We work hard, but at the same time, we devised these high throughput manners using this at least 96 well or the 384 well format. And already we tested about 100 analyzed. We made 10,000 compounds. 10,000 by 100 is the literally million, and 4 data points for different concentrations. We have millions of this data point, finding out specifically increase, decrease, and then find these sensors. Maybe too many data. We are too busy to write all of them into papers, and also boring. But what if the more challenging problem is, what if your sample unknown? You don't know what's inside there. Still, you want to detect something, especially complicated samples. So in the case, still there is some method you can use, so-called artificial tongue. Artificial tongue means our tongue has about 100 receptors, but we can discriminate 10,000s, 20,000s of different tastes by these combinations. What do you mean? What do I mean by that? So let's say rather than one sensor, you can use multiple sensors. We have many dyes. And then here you spike something and taste. Something you don't want to taste using your own tongue, let's just say cadmium. By spiking cadmium, this is before and after cadmium addition. Can you recognize which sensor has changed? There's some, right? But it's too many data, then it's not easy. So you can use still commercial other software, so-called Photoshop, and take two pictures and import and then let them generate different pictures. Then you can see, okay, this three has some color changes very quickly. And then you can convert this into digital info by taking out those RGB values. And then you can make up, okay, this is decreasing, this is increasing. How about the different one? So let's just say mercury. Still, you don't want to use your own tongue. And then many sensors are found there. And then if you generate these patterns, then you can get this kind of pattern. Now the idea is by collect those sensors, and then with the different concentrations, here aluminum, and depict this one into this kind of a pattern analysis. This looks like a voice pattern kind of things, right? Now the goal is using this pattern with the mercury patterns, whether we can distinguish them or not. That's the idea of artificial tongue. When I was in New York, we made this tongue, and then we called them as a New York tongue 1. And we tried to purchase every possible other metal cations from Sigma Aldrich. At the moment, we could buy about 43 or something, and then test them all. Interestingly, we could distinguish almost every other metals. There's a pH line outside here. By PCA analysis, you could see all different other metals here. Now, 7 years ago, 8 years ago, I moved to Singapore. And then I wanted to try, again, this one into the more practical problems. Again, Singapore water is quite the well-controlled water, good water. No worry much, but these tap waters. One thing was the tap water when I tested in New York. New York has 5 boroughs, Manhattan, Brooklyn, and Bronx, and Staten Island, and Queens. Each areas of water are different. And I tested again, hotel, hospital, and subway, their water is also different. So it was very hard to get the general idea. After I came to Singapore, I tested Malaysia, Indonesia water, forget about it. It's all different. House to house is different. Even that has the same problem. Korea, Japan, depending on those the different locations, the water are very, very different. Singapore, size-wise, quite good. You can have almost every positions. At the moment, we didn't have a Jukun area here yet. Up to the Boon-Lay, still we could get 60-something of this tap water from every stations of Singapore. It's a very good sampling, whole number of sampling, and then test them. Amazingly, this Singapore water is almost homogeneous. 90% of water is the same water. Only 5% of water is a different class. I was very, very wondered, and then called, I was not sure whether I can publish this or not, so I called the PUB, the water agency in Singapore, and they said, wait, we are coming in. And 2 detective-like people came to my office. Okay, I will explain everything to you. We are developing new water. Singapore has a short of water. We develop the recycling of those waters. But still, those waters may have some flavor, better flavor. That's why they don't use that water right away. They put them into the reservoir to renaturalize. Still, they're importing those water from Malaysia. And then 90% of water, only one check. And then using the pipeline, same water is distributed the whole Singapore, that's this water. 10% of the area, depending on those supply demand, they get this so-called new water. So that's why this is a little different class of water. Very interesting observation we found without knowing those facts. But they didn't stop me to publish this. If there was a journal of Singaporean chemistry, we could publish there. But so far in Singapore, there's no journal like that. So nearest the one was Journal of Australian Chemistry. So we published this work there. And then I found interestingly, this test, now these all waters are still safe water, new water or old water, it doesn't matter. But what if there is something funny? More important problem is whether this water is safe to drink or not. So that's why we tested this again. and then spiked with the different materials. And then again, this is the tap waters. And then combined the area 1, area 2, everything is inside here. That means that this water is a safe water to drink. Anything spiked here is outside of this circle. So this is a safe zone, and this is a non-safe zone. But we tested only one concentration. What if it is a variable of concentrations? Then dilute further and further, then it should emerge into this circle. So that's what we did. And everything comes to here. So this is safe area. Also, I started over here. It's something funny. You don't know what that is, but at least it has something funny, like a heavy metal or heavy metal-like materials in there. So using this concept, we wanted to provide very quick tests using this artificial tongue to solve this kind of problems. This is half of our work. More challenging problem as a chemist, biological systems. In human bodies, they say about 900 different cell types. And then to discriminate them, distinguish them, they use antibodies. Antibody number known to the human other cells, about 350 for cell surface markers. 900 different cells using 350 antibodies, can you distinguish them all? It's impossible. We don't have enough numbers of other antibodies. That means to define one cell, you should use 5 positive antibody, 3 negative antibody. Then you can say this is qualified mesenchymal stem cell or something like that. So we wanted to attack this problem using these chemical probes. Now, we don't know how they are different, but if you pick up one cell, so let's just say stem cell, and then select out most probable near the neighboring cell types, and then test them. And before you ask why they are different, what is the difference, that's a hypothesis-driven approach. But in this case, you just use a whole cell as a screening system. And this is the feeder cell, which is a helping cell around. And by adding same amount of dyes here, and we tried to find out and one compound which shows only one type of cells. Again, high throughput, high speed method is important. And this is our optimizer conditions. 384 well plate, this is size. And still, one other column, one well, can take 5,000 of cells. And you grow those cells line by line. Non-stem cell, stem cell together. And then second and third one has the stem cells. Then you want to see first column weaker, and third column and the second column is stronger. And those compounds has a potential to bind to these stem cells. So using this concept, we found this first embryonic stem cell compound. We gave those name as the CD, compound of designation, similar to the antibody names, CD yellow one. Second one, we attacked those neuronal stem cell, and this one we called the CD red 3. Same format. Embryonic stem cell is another control, and neuronal stem cell. And out of these 4 types of cells, now you want to see only these cells. Then by adding other compounds, we found, yes, only the neuronal stem cell can be visible, CD red 3. So this kind of work has been extended further and further. So embryonic stem cell, neuronal stem cell, one way is to make them by differentiation. And then by adding these dyes, okay, this is a purified, pure neuronal stem cell kind of things. One more example. In the brain, we have embryonic stem cell make a neuronal stem cell. And then in the brain, there are 4 different types of cells. And Wikipedia says it has 10% of the neurons and 10% of oligodendrite, and then microglia, and majority of the astrocytes. Look like they are different shapes. So I thought maybe you can see which one is which. But if you grow them, actually it's very difficult. It's messy. And dying cell, dead cell, and some contaminated cell, all different things in there. So 4 different types of cells, 4 different colors. Maybe you can show all of them. That was a very naive idea. It took 4 years and also one other graduate student's time. And we didn't find all 4, but so far we found 2. I have 2 minutes. And we found that one is the microglia, and then this is the neurons. And it seems like a snapshot, but actually it's not. It's live cells. Means that by adding these dyes, still you can stain them and they're moving around. And if you add this the microglia into the cancer cells, this green is a cancer. Now you can see this microglia attack these cancer cells and eat them. This is similar to the macrophage. Macrophage, our picture is one macrophage eat whole other cancer, but cancer is much bigger. Actually, they are sharing. So one cancer cell is eaten by others. How about the neurons? Neuron is much slower moving. But if you take those movies over 2 days, then now you can see this movement of the neurons. This is the cultured cells by adding the compounds, you can visualize this. This is the first small molecule probes which can do this kind of visualization. And then it can show those exon and other different structures moving around. Okay, I think I used most of my time. So we developed so far this kind of different other colors of materials: red, green, stem cell, mouse, microglia, and neurons, and all other different type of other materials. And we are moving to the cancer stem cell as the next the topic. But today it seems like I don't have enough time. Let me just skip it. And I have 2 labs. One is in medicinal chemistry program in NUS chemistry department. The other one is the government-supported institute in ASTAC, SBIC, Singapore Bioimaging Consortium. So there we have all those small animal imaging facilities, and then we can do PET and SPEC. So we work on this probe development, apply them into cells and tissues, and then using these colorful materials, but eventually to make this into the human version. Fluorescence cannot penetrate several centimeters, so that's why we need to have long wavelengths or SPEC or PET or the MRI. Then we can do small animal and also the human. And this is my combinatorial list of my collaborators. And always I'm looking for the new people that are working together with me. Thank you very much for your attention.